Okay, so today we will have our first lecture on the ChipSeq analysis. So in ChipSeq analysis, what we try to do is to understand the binding of certain transcription factors on the DNA. Just write it here, so the binding of transcription factors on the DNA. I must stress here that every time we are looking for a specific transcription factor, so we cannot just study the whole set of transcription factors in a certain moment. Instead, we focus on one transcription factor and we can assess where this transcription factor is bound on the DNA. So specifically today we will study the role of TAL1 transcription factor during hematopoiesis in mouse models. So our work will be based on this tutorial from the Galaxy, and they have used a data set from a study in mice. Now, this TAL1, this protein that they study, it seems that it plays an important role during hematopoiesis, and what they have done is that they have used different type of cells, G1E, which are precursor cells, a bit undifferentiated, and megakaryocytes, which appear later in the differentiation. What they have seen is that during differentiation, the location of TAL1, so the genes that the TAL1 regulates, has changed dramatically. Now for us, it's not so important at the moment to study the differential binding of TAL1. Instead, we will focus only on these cells, on G1E cells, in order to see the technical details of ChipSeq. Okay, so what we will do is that we will go to Galaxy, and first we will upload the data into Galaxy. So in order to begin, we will make a new history, an empty one. I just have uploaded the datasets here. So an empty history here, I called it ChipSeq analysis, and in order to upload the data, we go here and then we select paste fetch data. So it's waiting for us to upload the data. We can put a name here, TAL1 or whatever, because the data will be from TAL1. And here we will put some links. So from the tutorial, it provides a link here in Zenobo, where usually researchers upload data for open access for everybody to use. So I must mention here that this dataset here is not the whole dataset. They have downsampled, which means that they have taken less reads than what they have in the publication in order to speed up the analysis for this tutorial. And this is what we will do as well. Okay, so if we go here in this link, we can download these 4 FASTQC data. It's just FASTQ files. In principle, they just have named it FASTQC. It's not important for us. So these are 4 files. 2 are with immunoprecipitation. So these are the treatment files, and these 2 files will be used as controls respectively. So the control for this TAL-R2 will be this input R2, and the control for this TAL1R1 will be this input R1 file. Okay, we can just copy the link and then go to Galaxy and just paste the link here and do this for all 4 files. Then we will just press start here and the data will be downloaded as we see here. Now if we view the data, these are the data, and then we can proceed as we have done already with FASTQC and so on. So I just click here FASTQC, and here in the multiple files I will just use all of them. Have no contamination and no adapter list here. I hope it will find itself if there are adapters, and I continue. So this is the results from FASTQC. We see that the data quality is not bad actually. Maybe here in the end a little bit some reads needs trimming. So the next step in the next video will be the trimming. Instead of using the SQL software that we had used in previous times, now we'll use a Trimomatic just to follow the tutorial. Exactly, so they use here Trimomatic for trimming, and in order to learn also how to use a new software for trimming.